Hello, everyone, and welcome to today's session. I am Padma Natarajan from CCBB, the Center for Computational Biology and Bioinformatics here at Scripps, California. Today's session is the second of the two-part webinar series on pathway analysis hosted by CCBB. Once again, our webinar presenter is Dr. Cordelia Ziraldo, Chief Science Officer at Advaita Bioinformatics. Dr. Ziraldo leads product development and technical support for Advaita's state-of-the-art bioinformatics platform. In the first webinar held last Tuesday, Cordelia focused on the overview of iPathwayGuide, the pathway analysis software. This is their flagship product, and we learned about the importance of having an up-to-date knowledge base in the interpretation of your omics data. We also know how pathway analysis is done in Advaita in comparison to other similar tools. And we also know that Advaita generates an automatic report generation and also data sharing ability available with Advaita's iPathwayGuide. Today's session will be a deep dive session on some special aspects of iPathwayGuide. Before we begin today's webinar, I would like to make a few announcements. There will be a Q&A session at the end of the webinar. We would love to hear from all of you. As before, a recorded version of the webinar will be available to all the attendees. I also would like to remind everyone that CCBB has institute-wide license for iPathwayGuide for unlimited number of analysis. So please email ccbb@scripps.edu for any analysis requests. For those of you joining just now, welcome again. With that, I request Cordelia to present the webinar. Today's topic is get the most from your omics data, maximize your pathway analysis with iPathwayGuide.
Thank you so much, Padma, and thank you all for joining us this morning/afternoon, depending on which coast you're joining from. The goal today is really to give you a closer look at this data analysis platform and what it can do in order to help you think about how this platform can be useful to you as you analyze your own data. A little bit of background for those of you who didn't join us last time. I want to make sure we're all on the same page. Our leadership team is comprised of our founder, Sorin Dragic, who has a PhD in computer science and also has more than 20 years in bioinformatics. Most of his career has really been focused on the analysis of life science data, including 10 years as a bioinformatics core director. He's currently the co-director of the Bioinformatics and Statistics core at Karmanos Cancer Institute. My PhD is in computational biology, and I have more than 10 years of what I call collaborative data analysis. This is working hand in hand with life scientists to help them analyze very large data sets. But we noticed a problem, which is that when the interpretation of biological data is left up to bioinformaticians, a really important component gets left out, which is the life science domain expertise. So in order to solve that, we realized we needed to create a platform that would allow the life scientists to really dig in and interpret their own data without getting muddled in the math. So that is why we have created iPathwayGuide. And here are just a few of the benefits for researchers. The first one is that it's highly secure remote access. This is a cloud-based platform, which means you can access from anywhere. You can receive your results, a link to your results by email, and you log in through your browser. And whether you're at home or at the office, or one day when we start traveling again at a conference, you'll be able to log in and interact with your results. This is really focused on empowering the domain experts to be the ones to make extraordinary discoveries, because you'll be able to use your in-depth knowledge about the condition to guide your interpretation. You will not have to wait for a bioinformatics team. You'll receive your results from the core, but then when you have a question or want to dive deeper, you'll be able to do that yourself right in the platform. Instead of sending an email that says, oh, could you send over the next 5 results? You gave me the top 5. I'm curious about the top 10. And waiting for your bioinformatics team to have time to get back to that request. This is much more streamlined interaction. And it also means that your results are guided by and focused on the biology rather than produced by mechanically applying standard pipelines. We provide an audit trail so that you always know what versions of the databases were used for every single inference. And you can always go back to older versions as needed. There's easy and unlimited sharing, which means you can forward your results on to a collaborator across the world and ask them to weigh in. There's no license required for people who accept these shares, which means anyone, really anyone, can collaborate with you. And it's very simple. It's a single click and you enter their email and they get full access. And the last thing is there's an automatically generated PDF summary of every analysis. It's a comprehensive 25-page research report. I've seen people use it as supplementary materials. I've seen people use it in the method section of their manuscripts. And it's just a really nice, full featured explainer of the top results from your analysis. Okay, I want to give you, people ask us what's different between us and the competition. And I think the most important thing is that the platform is really guided towards understanding the results, finding the story in your data very simply without, spending hours digging. And what I mean by that is, here's a picture of a gene network. This is a figure you would see commonly in a lot of papers. There's a lot of things happening in this figure, but for me, it's really hard to get any real information here. There's just, it's impossible to read. There's a lot of genes connected to other genes, some more closely than others, but I'm really not sure what can be said here. On the other hand, what if you could produce a figure like this, where you show these are the genes that participate, and here's the mechanism through which they influence this process, a process that was important in my experiment. And the gene that I've highlighted here controls the rate limiting step towards this process. This is a story. It's simple to understand. It's easy to see how the conclusion was made. This figure was created in iPathwayGuide, and today I'm going to show you how to do just that. The second thing that differentiates us from the competition are the algorithms that we use. The platform was based on impact analysis, which is an algorithm that came out of Serene's lab and has since been cited more than 1,500 times. But we've extended that philosophy, and I'll tell you a little bit about impact analysis in the next slide and how it works. But we extended that philosophy into our sophisticated gene ontology analysis, as well as our proprietary mechanism inference engine, which was what was used to create the figure on the previous slide. So that you can ask questions like, how do my differentially expressed genes trigger this observed biological process? And lastly, we also offer drug repurposing predictions, which I'm going to show you as well. Okay, so I want to just give you a side-by-side comparison of enrichment, which is the standard way that people generally do pathway analysis, versus impact analysis, which is what's implemented in iPathwayGuide, just so you have an appreciation for how we approach pathway analysis. So from an enrichment point of view, let's look at these two pathways. They both have about the same number of genes on them. And we're going to assume for this example that there's exactly one differentially expressed gene, and that's the insulin signaling receptor. So if you were to calculate an enrichment score where you ask, are there more genes differentially expressed on this pathway than by chance? You would look at the number of genes differentially expressed on the pathway, 1 out of the total number of measured genes on the pathway, about 40. And you would get about the same enrichment score for both pathways, and it would be probably not significant. However, if you look at the topology of the pathway, you'll notice that there's a big difference. In the adherence junction pathway on the right, you can see that there's nothing downstream of the insulin signaling receptor. So there's no predictions that can be made about changes on the pathway based on fold change in insulin signaling receptor. On the other hand, in the insulin signaling pathway, you can clearly see that there are several genes directly downstream of the insulin signaling receptor and other genes directly downstream of them. So you can predict that there will be changes in several of the pathway genes just based on a change in the insulin signaling receptor. So the topology score for the insulin signaling pathway is much stronger than for the adherence junction pathway. And for that reason, impact analysis would score insulin signaling pathway to be much more significant. So that's the difference. Last week, we showed some results comparing these two methods and showed that the accuracy of impact analysis beats enrichment. You get more relevant pathways a much higher percentage of the time. So if you're curious about the details of that, I encourage you to go back to the recording of last week's webinar and watch. Okay, here are some common tasks that people do when they have, transcriptomics data that they want to interpret. And they can do this also with proteomics data, also with epigenetics data. But here are the kinds of things that people usually want to answer. The first one is, I have a gene that I'm really interested in. I want to know what happened to it in my experiment, and I want to know what else is known about it. What pathways does it fall on, etc. The second is, what pathways are impacted in my experiment? Are there any affected biological processes, cellular locations, or molecular functions? What are the underlying mechanisms driving the results that I'm seeing? Are there any important upstream regulators, be they genes or chemicals or toxicants or drugs? Are there biomarkers that could be used to differentiate between my conditions? Or is there a specific time point wherein a biomarker will signal that a change is about to begin? On that note, can we perform a meta-analysis across several different omics types, or across a time course, or across a dose response curve? And lastly, people like to share their results and collaborate on the interpretation. So just one quick poll today, which is, which interpretation tasks do you do most often, and which are most important to you? You can vote for as many as you like. Okay. These results are pretty interesting. So all of you are interested in pathways and Go terms, or almost all of you, I should say. And a very strong majority of you also wants to elucidate underlying mechanisms. But half of you are interested in upstream regulators and biomarkers, and a quarter of you are interested in meta-analysis of several data sets. So what I'd like to do is take you through all of these and show you how they fit together as well. Let me hide the results and go back to the slides. Okay, so the plan today is to show you how first to perform meta-analysis. And I'm doing this first because it allows us, I think, a nice entry point into finding biomarkers as well as looking at impacted pathways. And you'll see what I mean when we get in there. And then after that, we're going to look for affected Go terms, identify upstream regulators, and then lastly, look at this underlying mechanism and really how to build those network figures. Okay, so we're going to jump into the software. The first demonstration data set that I'm going to use is the analysis of 4 breast cancer subtypes, each of them versus healthy. So there's a triple negative group versus healthy. There's a HER2 positive group. There's luminal A and luminal B. And we're going to look at the meta-analysis of these. The data comes from Geo. It's a public data set. We are not affiliated with the authors in any way. Okay. So you can still see my screen. This is iPathwayGuide. Here is our meta-analysis of 4 breast cancer. Okay. This is the summary page. It's the summary for the first contrast, triple negative versus healthy. So this is a summary of the input data and the results for that first contrast. And there are 4 of them, so you could see the results from any of these. But we're going to go straight to meta-analysis. Okay, here we are in the Genes tab. You can see across the top the different modules of the analysis go from simplest to most complex. And here what we are looking at is a Venn diagram comparing the genes that are differentially expressed in each of the 4 contrasts. So the way you read this is that there are 706 genes that are uniquely differentially expressed in triple negative. And there are 519 genes that are commonly differentially expressed across all 4 contrasts. And you can see that this table here has been filtered. When I make a selection, it's filtered to include only those genes that I've selected. And you can also see that the color here of the number corresponds to the fold change. So a blue fold change is negative, meaning that the gene was down in the condition compared to control. And a red number here means upregulation. So what we can do, a really quick way to find biomarkers, is simply to sort this table by fold change. And we're going to sort by triple negative because we're curious about a biomarker that would uniquely identify the triple negative condition. And we're just going to open this up and scan with our eyes. We're scanning for a change in color. And almost immediately, we find ESR1. This is down in triple negative and HER2. It's up in luminal A and B. ESR1 is the estrogen signaling receptor. It is the canonical biomarker for the luminal subtypes. So this is heartening to see. In fact, you can see it here. But it does not uniquely identify triple negative. So we're going to keep going. And as we keep going, we quickly find CA12. And here is a gene that is uniquely down in triple negative and up in the other subtypes. So it is unique, it has a unique pattern in triple negative. All of this literature here is specific to the gene that's selected and very up to date as well, very current. So another way to find biomarkers is to look for changes on pathways. So you can do the meta-analysis across any module. So let's look at pathways. And now you see here, we're looking at significant results across contrasts. So after FDR correction, there are 19 pathways that are commonly significant across all 4 subtypes. And we see them here in this table. We can also look at the rank diagram, which allows us to see which pathways are more or less significant. You know, how does the rank change here? Let's look at the Pixary signaling pathway. And when we look below, what we have 2 things. We have the pathway diagram, and this shows you which genes are differentially expressed in how many contrasts. So the color of the green, the intensity of the green in this pathway map corresponds to the number of contrasts that have that gene as differentially expressed. So for example, RHEB is this gene right here, and you can see that it is differentially expressed in all 4 contrasts. But this one, on the other hand, is only differentially expressed in 1, and it has a much lighter color to it. So we can also use this gene bar plot as another way of looking for biomarkers. And we're going to do the same thing, which is to scan for a gene where we see a change in the direction of the fold change. So really just quickly scanning by eye. Here's an integrin that is down in luminal B. It's up in luminal A and HER2. It's not significant in the triple negative subtype. So that's a possibility. The other thing we can do is look at the pathway diagrams themselves and see how the pathway dynamics shift across the different subtypes. So we can look at this Pixary signaling from the perspective of triple negative versus healthy. And we can look for coherent cascades. So this is just fold change, and we're just looking at where do we have agreement between what's on the pathway and what's given in our data. So where do we have interactions between genes that match the fold change patterns that were measured in this experiment. And we can step through and notice where those coherent cascades change from contrast to contrast. And you can see that there's some things that remain the same, there's some things that get extended further, and there's some things that change. This last button here, auto cycle, allows you to do it without clicking. So we see something goes up here, this ECM goes up and then down. I think this is a nice quick way to get to start generating hypotheses about where is the crux of the difference between our contrasts. And when we find something that we're really interested in, we can go and dig into it a little deeper in the contrast itself. So let's look at this Pixary signaling in the HER2 pathway. the Pixary pathway in the HER2 contrast. Okay, so we are viewing the fold changes for this specific comparison on this pathway. And you can see just as before, blue means downregulated, red means upregulated. And we can look at this a couple different ways. We can actually look at not just fold change, but accumulation and perturbation. And these are the topological signals that allow us to see how these fold changes are propagated across the pathway. So just like we showed in the slides when we were comparing the insulin signaling pathway to the adherence junction pathway, here what we can do is propagate the fold changes according to the interactions on this pathway. So this is what we have for fold change, and what we're going to see with accumulation is how these get propagated. And then the perturbation is going to put those two things together. So where do we have agreement between what's measured and what's predicted, and where do those two things clash with each other? And then Coherent Cascades will show us really where our putative mechanisms are. So we have a really strong chain here going all the way to MAP kinase. We also have a lot of activity going into AKT and then out to the cell cycle processes as well. Another nice thing that can be done is once you start focusing on specific parts of the pathway, you can ask, are there drugs or microRNAs that target these genes? Because I want to test the hypothesis that this is really changing. So we could select Pixary, for example, and yes, there are drugs that target this gene. And when I select them, their targets are highlighted here. So you can find reagents essentially doing things like this. Okay, we're gonna go back. There's a ton more you can do here, but let's keep it moving. So we have performed a meta-analysis. We have found biomarkers. We've identified impacted pathways and even started looking at mechanisms on those pathways. So next I want to look at biological processes, identify important upstream regulators, and then really spend some time looking for underlying mechanisms and elucidating those mechanisms. So we're going to use a different demo data set for this part. This is an osteosarcoma cell line that was treated with a synthetic mirror, and one group got a sham mirror instead of the mirror. 48 hours later, RNA was isolated. This is again a public data set. We have no affiliation with the authors. I want to show you the paper to point out two important findings. Firstly, that P53 is positively regulated by this mirror. And secondly, that the ultimate result was that cell proliferation was inhibited. Okay. So let's go back to our dashboard. These are both demo data sets that are available to every single person who registers on the platform. So if you already have an account, these demo data sets are available to you. Okay, so let's look at genotology terms. And we covered this a little bit last week, but I want to take a deeper dive and talk a little bit about why genontology can be tricky. So if you look here, what we have with the standard genontology result is 156 pages. So 1,560 significant results. And if you just start going down the list one by one, you're going to be here forever, first of all. And you're going to see that the top results are repetitive. Cell component organization or biogenesis, cell localization, cell protein localization, organelle organization. So I'm repeating myself, and it's really unclear whether these are all part of one important mechanism or if there's a couple different processes that are important here. So the first thing you can do, and this is available in every module, is select Bonferroni correction. This is the most stringent correction for multiple comparisons that we have. You're still at 150 significant results. But then you can also look at Dendrogram View, which is a slightly different view. So notice that this wheel goes from pink, which are the most significant, all the way around to aqua, which are the least significant but still significant terms. In the Dendrogram View, I'm going to actually make my window smaller, ironically. Okay. So here in the Dendrogram View, you can see that there are actually a couple different pockets of bright pink. So what we have are, these are all of the significant terms according to Bonferroni, and they're arranged so that terms with many common differentially expressed genes are close together. So this is based on the differentially expressed genes in the input data. So for example, regulation of cell motility is right next to regulation of locomotion. And if we hover on this white node here, you can see that these two genotology terms have 103 differentially expressed genes in common. Now, they may have more than that in the ontology itself. But again, this is based on the differentially expressed genes in this experiment. So we have cellular component organization is the one selected. That's why it's yellow. Next to cellular component organization or biogenesis. And you can see that these two Go terms have 525 genes in common. And as we move up the tree, we collect more and more terms together. These 10 Go terms have 36 genes in common. So the question is, how similar are these to this little pocket over here? So what we have over here is the localization terms. So we have this component organization, and these are cellular localization, macromolecule localization. So to connect these, we would have to go all the way to this node here, and these 67 Go terms have 0 DE genes in common. So that tells us that there are at least two things happening here that are actually disparate because they're not based on any common differentially expressed genes. But we can do better than that. They're still not super specific. We're still not really getting into the information that we really want. So we've implemented two different types of pruning factors that allow you to get to the most specific Go terms in the hierarchy, the ones that are at the very bottom of the tree, that are still significantly related to your set of differentially expressed genes. So we'll select high specificity pruning. And now we're down to 3 significant terms instead of 150. And you can see that the first one is DNA damage response signal transduction by P53 class mediator resulting in cell cycle arrest. And you remember that paper said that P53 was activated and that cell proliferation was inhibited. So the third one is cell proliferation inhibited, negative regulation of cell population proliferation. But you can just see that we're just way more specific. This is an actual, this is a biological process that tells us something about what's going on in these cells. Okay, so that's really how to get at some nitty-gritty information about the biological processes. The same exact thing goes for molecular functions and cellular components. Let's look at upstream regulators and chemicals. So we have a database of interactions between genes, and some of those interactions are directional. So A is upstream of B, and A activates or inhibits B. And using that kind of information and setting the differentially expressed genes as the target, so playing the role of B in that example, we can calculate and predict genes that would be active or inhibited upstream of the differentially expressed genes, and use the direction of their fold changes to make those predictions. So when we look at upstream regulators that are predicted as activated, the top upstream regulator that follows that prediction is TP53. And that was the title of the paper. You can see that in the input data, TP53 itself was not a differentially expressed gene. Its fold change is barely above 0, and its adjusted p-value is not significant either. However, there were a large number of targets of TP53 measured in the experiment, 271, and of those, 55 were differentially expressed, which is a pretty big fraction. And of those 55 differentially expressed targets, 48 of them were consistent, so changed in the direction that matches the hypothesis that TP53 is activated. So only 7 contradicted that hypothesis. And you can see them here. What you should notice is that when we have an inhibitory relationship between TP53 and a gene, that gene is downregulated. And when we have an activating relationship between TP53 and a gene, that gene is upregulated. So that's how we come to the conclusion. That's how we predict that TP53 is activated, because the direction of these changes matches that pattern. If you're interested in any of these specific interactions and want to know how we came to that conclusion, there is a table below with full evidence for every single edge. And you can look at each interaction and see the score. This confidence score goes from 0 to 1,000, and it tells you how strong the evidence is for that interaction. There's links out to other databases, and when there are papers to help bolster the evidence, we offer them here with links to PubMed, DOI, and PubMed Central as well. You can see that the genes are also highlighted in the title and abstract when they are mentioned. Okay, so this evidence table is a really nice way to inspect evidence in the network when you're not sure. You can also predict upstream regulators as microRNAs and chemicals. With microRNAs, we won't spend a lot of time, but I just want to point out that when you look, even with Bonferroni correction, the #1 top result for this data set is Mir 542-3P. And if that name sounds familiar to you, it's because that was the Mir that was used in this experiment. That's the reagent itself. So just a proof of concept. We're predicting microRNAs that could be active based on downregulated DE genes. And these are the targets right here for this microRNA. Okay. With chemicals, similar to upstream genes, you can look at chemicals that are predicted to be present. So this is a chemical that if you if you used it, would mimic the phenotype that you're measuring, that you're observing here. But you could also look at chemicals that are absent or insufficient. And these chemicals, the way I like to think about this is these are chemicals that could rescue the phenotype because they would reverse the differential expression that's seen here. So, for example, we have adenine, which is it's a purine base, right? It's part of nucleotides, but you can see that it has 22 consistent targets. So these are all targets of adenine that normally it would activate, or sorry, it would inhibit, but all of them are up. All of them are upregulated. So adenine would reverse this upregulation if we added it to the system, essentially. Okay. Let us go to networks. Let's start putting this all together. Okay. So this module is just a little bit different in terms of how it's laid out than the other ones. And the idea here is that you are given the tools that you need to explore pairwise interactions between genes of interest, and also ask, how are my genes of interest driving this biological process that I'm observing? What chemicals are affecting my genes of interest? What's the mechanism? And you can even go as far as to say what mechanism connects this drug to this process, right? So if you were to take all differentially expressed genes in an experiment and throw them into a network and ask, how are they connected? You're going to really quickly get a hairball that you cannot read or interpret. So we've set up some tools to help you filter your set of genes down to a manageable number of genes of interest. And here's the workflow that we recommend that you do. Step 1, use your analysis results to select the genes of interest. So you could say, for example, we know based on the results that TP53 is really important here, right? So what if we say, let's look at the targets of TP53, since we know that they were, we know that it was activated. And let's ask, what do these have in common with our result about, cell proliferation being inhibited? So we go to our biological process. So I'm now even in the biological process section, and I'm going to apply the high specificity pruning, and I'm going to select negative regulation of cell proliferation. And you can see that what happened is we found an intersection. So these genes here are exactly the genes that are targeted by TP53 and annotated to negative regulation of cell population proliferation. So this might be interesting to look at. Let us then do one more thing, which is let's add TP53 into this mix. We know it's not a differentially expressed gene, so it's not going to come up in step 1. But in step 2, you can fine-tune this set of genes here by adding or subtracting genes from your selection. So now we've got TP53. And if we then click build, we will see what's known about how these all interact. And the answer is TP53 is obviously known to regulate all of them. but we don't actually see any other interactions between any of these other genes. So the next thing we could do is add a process here. We could say, and how are these specific genes related to our most important biological process, which was DNA damage response? And here's the mechanism through which they influence that. It's specifically these 4 genes: CDK, BTG, BAX, and TP53 are the genes that mediate this DNA damage response. You have a couple of options here. So you can look only at regulatory interactions. When I select this box and I click build, we have removed, essentially we've removed binding and catalysis and reactions, because those are the interactions that are bidirectional, essentially. So these are all, you can see the flow of information more clearly this way. So this is nice. We can clearly see TP53 is regulating all of these. These are the genes that are mediating this process. The last thing I might do is add in a chemical and ask, we saw that adenine can reverse the phenotype. Are some of those genes here? Are there reactions between adenine and any of the genes in this mechanism that could be used to influence this process? So up here we can add there it is, and I click build, and the answer is yes. 2 of those 4 genes, hold on, you can't quite see. So adenine inhibits BTG and CDKN1A. So it essentially could reverse some of the things that we're seeing here. There's all kinds of mechanisms that you can build. And there's all different ways to interpret the network. So let's try another one. Let's look at, I'm going to clear. Let's look at the first pathway result is P53 Signaling Pathway. That fits in very nicely with everything else that we've seen in this data set. The next one is pathogenic E. coli infection. Maybe we're curious about why that's coming up as significant. So we can select the genes on the E. coli pathway. And so we've got 32 significant genes here. And I'm going to leave this DNA damage response go term in the network. Let's ask the question, how are these 32 genes influencing this biological process? What does that mechanism look like? And I'm going to increase my confidence. Okay, so what we have here is that there is exactly 1 gene on this pathway that influences that mechanism, and there are a few other things going on. You can hide the isolated nodes so we have a clearer view. So now maybe we want to say, okay, I see that we've got a couple different mechanisms happening, but the question really is, how are they connected? So in order to do that, what we're going to do is include intermediate genes and ask the network to sort of fill in the missing connections. We'll start with 1. Okay, so it was able to find differentially expressed genes that connect all of these mechanisms together. Interestingly, this gene connected to this Go term stays by itself. So the answer is this really isn't connected. This pathway is regulating a different part of the story, but not this DNA damage response. This is how I interpret it. Okay. You can do this kind of network analysis with all kinds of different things. I want to show you other ways to interpret the network. So let's stay with this network and look at a couple of different views. Above the network diagram, there are several tabs, and each of these tabs is a way to view the information. So subnetworks helps you to see where you have different separate networks. You can see you've got one here and one here. This subnetwork is a highly interconnected group, so it's really everything is clustered together. If you had, in this connected group, if you had one part that was more tightly interconnected than the rest of it, it would be a little bit more separated. The Mechanisms tab allows you to see a simplified view of the network. It tries to maintain all of the connections. So you can see you don't lose genes that are connected to other genes, but it gives you a simplified view of this in order to help you look at mechanistically what's going on. And here you can highlight coherent cascades. So just like on the Pathways tab, these are places where you have agreement between the directional fold change and the directional interactions between genes in the network. Hub genes is the default layout. These genes are arranged so that the ones in the center have the highest number of connections, of neighbors, and the ones on the edges have the fewest. Gatekeepers sort of emphasizes the places where, if you are, I'd like to think of them as bottlenecks, essentially. If you're passing a message from one end of the network to another, the gatekeepers are the ones that are almost certainly going to be hit. Okay. Interactions lets you see where you have a concentration of edges. And the Regulators tab arranges things vertically according to the flow of information. So if A is upstream of B, then A will be above B, essentially. Okay, it's 3:25, it's 12:25 on Pacific time, and I want us to have time for questions. So I'm going to pause here and let you guys ask a few questions before we end. You can put them in the question section. Maybe I'll start us off by asking Padma, can you please remind us one more time, how do you get access? What does it cost to get access to iPathwayGuide? How long does the process take? Those kinds of questions.
Yes, we are happy to do the iPathwayGuide analysis for anybody at Scripps. All you have to do is basically make a request to ccbb@scripps.edu, and then I will be able to send some example file formats in which you can give your data for uploading onto Advaita platform. Then after analysis, I will be sending the links out to you. You should have an account on Advaita in order to access those results. and you will be free to share these results with anybody you are collaborating with, whether they are at Scripps or outside of Scripps. All they need is an account on Advaita, which is for free for registering on that platform. So the charge model and example files, all those will be sent out to you once you make a request to ccbb@scripps.edu. The charges are very nominal, and sometimes they are built in the sequencing activity. So if you sequence your data at the genomics core here at Scripps La Jolla, some of those costs are already built in, for example, for RNA-seq analysis and things like that. So I can get back to you with more details once you send the email to me. Thank you.
Awesome. Thank you. Serena, are you seeing any more questions come in?
No, I don't have any questions at this point.
Oh, I see one. I see a couple. Can we import the GMTs from GSCA and perform the analysis? For example, if I want to look at hallmark pathways, GMT. I do not know what a GMT is. Are those transcripts? GMT, like differentially expressed genes? Can you, Aishwarya, can you clarify what that is, and then we'll answer this question? Okay, another question is, how do you import data? So this is where CCBB comes in. This is
yeah, basically we are taking the input in two different formats. One of them is a CuffDiff format, and the other one is a custom format with just 3 columns of data in it: gene IDs and log fold change and adjusted p-value. These are the 3 columns that we need for the custom format. So basically we are accepting in these 2 formats. And I can send out the example files along with the charge model and all the details once you send me the email to ccbb@scripps.edu.
Yeah, we have a lot of, between the two of us, we have a lot of resources for helping you get your data analyzed or formatted so that it can be analyzed properly. Okay, so I think what the question is about GSCA, I think it's about importing gene sets essentially to compare to your results. This is something that's actually in process right now. We are developing new capabilities that will include something that allows you to look at a specific set of genes compared to your DE genes. It's not available yet, but it is coming soon. Can you analyze mutated genes? What's the difference in downstream signaling and gene expression? Yes, you can. There's a couple different ways to do that. One is we have a second platform called iPathwayGuide that allows you to look at mutations. And the other way is that you can roll up your mutations into a set of genes, you can use that to analyze the mutations as one data layer, look at your transcriptomics as a second data layer, and combine them, creating using meta-analysis to look at where you have influence from one to the other. Okay, yeah, so Alan just sent me another link about gene matrix. Can we do all the analysis options presented today by our own on the platform? Yes, everything we did today is available to you. Once you get your results from CCBB, you will have access to exactly the interactive format that we went through today. So everything we did today was on what I call a shared analysis, which is what you'll be working with. All of that is available to you. And we do not, when you have a subscription, when you have a purchased analysis, every single module is available to you. So there's no There's no other version of the subscription that gets more modules. Every person has access to all modules.
So Cordelia, correct me if I'm wrong. So when you upload the data to Advaita, we use certain cutoffs on log4 change and adjusted p-value. Once those numbers are set in, so when we share the results with any of the researchers, they are not able to change that.
That's right.
By themselves. So if you request for a different cutoff on these two parameters, then we have to re-upload the data and it'll be a separate analysis again.
Yeah, every time you change those parameters, you change your set of differentially expressed genes. And then every single calculation, every single statistic that was calculated has to be redone because you're working with a new set of differentially expressed genes. So that's why it's a whole new analysis. But all of the things we did today, right, are downstream of that once you've submitted that set of DE genes. So all of that's available. Okay, we are a couple minutes over, so I don't want to keep going, but we're happy to continue answering questions. You know, you can get in touch with us at the emails here, either me or Serene or Padma, and I'm happy to continue the discussion offline. Thanks, everybody.
Thank you for attending. Bye.